can be turned on. What is the state, normal state of an inducible operon? Off. A repressible operon, what is its normal state? On. The two examples that I talked about was the lac operon, which is normally turned off, and the trip operon, which is normally turned on. Okay? And so, with the lac operon, it's turned off. Why is it turned off? The polymerase is blocked from what?
You said the last off-on is off, from the off?
Yes.
Yes, and you asked me why it's off.
Yeah, why is it off?
Does it have to do with the regulatory gene?
Could be.
Or is it because there's no lactose that binds with it?
True.
So that means there's no lactose in the environment?
Since there's no lactose in the environment, this operator is turned off, meaning that there is a suppressor that is on there. Okay? And the thing is, this regulatory gene here, right, is producing the suppressor. Right?
So it's turned off because there's what on the?
Suppressor. Suppressor. Okay?
Because there's a suppressor, that's why it's turned off?
Yes. So this would be your LAC Z, LAC A, and what's the other one? LAC Y. Now these are the genes right here. And what do these genes code for? These genes are going to code for what? A protein, right? And that protein is going to turn into what? An enzyme that breaks down lactose. But it's turned off because right at this moment, there is no lactose in the environment. Now what happens when lactose is in the environment? It's going to have to get turned on. So, now you have lactose that is in the environment that will bind onto the suppressor. So what happens when it binds onto the suppressor? So now your suppressor
Guess the polymerase a quick shot.
comes off of there. So yes, it gives the polymerase. Now you have the polymerase, which will bind onto here, and then be able to read those genes. Okay? So this is lactose here.
So the polymerase
Huh?
binds onto what?
Now here you have your RNA polymerase, right?
Okay, wait, so lactose is usually off because there's a suppressor when there's no lactose in the environment, but when lactose is there, it binds onto the suppressor, allowing.
Yeah, it binds onto the suppressor, causing the suppressor to change shape. If it changes shape, it will no longer bind to the operator. Now that's going to allow the RNA polymerase to come through. As it comes through, now you're going to get transcription here, and then you're going to get translation and you'll get your protein. Which will become the enzymes that will work on the lactose. Okay? So, normally this is turned off, right? That makes it an inducible operon, right? And this is the lac operon, which means that its job is supposed to make the enzymes that break down lactose, okay? But since it's turned off, that means that there's no lactose in the environment. Now I take this bacteria and I put it on an EMB plate or McConkey plate that contains lactose, right? I put it into the incubator, and 18, 24 hours, I can tell whether or not this lac operon was turned on or turned off, right? If it's not turned on, or maybe the bacteria doesn't have that operon because it doesn't utilize lactose in that fashion, then it will be lactose negative. When I looked at the bacteria that was lactose positive, obviously that lac operon was turned on because of the presence of lactose. The presence of lactose will bind onto the suppressor. I'll put it here. Binds onto the suppressor, not allowing it to come onto there, right? Since the suppressor cannot come on, the RNA polymerase is now allowed to read through and express those genes. Through transcription, you get your RNA. That RNA is then translated into the proteins, and those proteins will eventually form the enzymes. Now those enzymes will be able to metabolize that lactose that's now in the environment until it's all gone. And then when it's gone, this will be suppressed because there won't be any more lactose in the environment to stop the suppressor. The suppressor will jump on there, stop that from working, and it may go on to the next carbohydrate, which could be arabinose or some other type of carbohydrate that it could use, or it may die because there's no more food in this environment. Okay, so that is your lac operon. That is the inducible one. Yes?
So after the lactose is binding to the suppressor, causes the suppressor to change shape.
Yes.
What's after that?
The suppressor changes shape. It can no longer bind up to the operator. Therefore, it can no longer prevent the RNA polymerase from reading DNA.
It can no longer read DNA.
It can no longer stop the RNA polymerase from reading the DNA. Therefore, it's going to be read, it's going to be expressed, it's going to be translated, it's going to be made into the enzyme that will then be able to work on the lactose in its environment. Now, let's look at your repressible ones.
That's still the number 9?
Huh?
Yeah, those are for years.
So, once again, you have your,
The repressible ones.
So this is the trip operon?
yes.
Okay.
So, you have the same thing here. You have the operator. You have a promoter. And you have your regulatory gene. Right? But this one is normally turned on. And is normally turned on, and it's going to be producing tryptophan, right? So,
How do you spell it?
okay. So it's being transcribed. So you get your RNA. And you get your protein from it. Okay, so now you get your protein. And that protein, or here in this case, we're talking about the amino acid. The amino acid tryptophan.
It's being transferred into RNA and then what?
It's being transcribed into the RNA and it's producing tryptophan, okay? So now you have all these molecules of tryptophan, and they're accumulating. Okay? So normally, this is producing an inactive form of this.
So usually for
It's producing it, but it's inactive. Okay?
the amino acid tryptophan.
This right here is a regulatory gene. The regulatory gene is producing an inactive form of the suppressor.
So it's usually producing an inactive form of it?
Yes. It's producing inactive form of the suppressor. So this would here would be your promoter. And this right here would be your operator. Okay? So this is inactive. But remember how we talked about the feedback loop? When you have an overproduction of something, it's going to then act as a allosteric inhibitor. So this tryptophan that is being produced in mass amounts, now it's too much, it will start to come over here and bind onto the inactive form here, converting it into an active form. Notice there's a change in shape there, right? And so this active form will come up to here and bind onto the operator. And when it binds up to the operator, it does not allow for that gene to be expressed. Okay.
So, wait, then after you said it combines with the inactive one and it turns it into an active
Yes, now it
suppressor,
is,
and then what?
this one here is now active.
To an active form, and then what?
Now the active one will bind onto the operator, right? The operator that has been bound, so now your RNA polymerase will not be able to bind onto there, and you will stop transcription. You see that? Tryptophan is being constantly made because this is on all the time. Well, when that tryptophan is made in abundance and too much is made,
So the buildup
it will
of it.
bind onto the inactive suppressor, causing it to change shape. It is now active. In its active form, it can bind onto the operator. When it binds onto the operator, it prevents the RNA polymerase from binding onto the operator or reading through on the operator because it's going to bind on the motor. Anyways, it may bind onto here, but it will not be able to read, so it will be kicked off.
So it prevents the,
It prevents the transcription of that gene. When it prevents the transcription of that gene, none of this stuff happens.
Okay.
That's
Question,
fine.
you had this line, the protein, you had the amino acid tryptophan, why?
This is always turned on. Its job is to produce
Okay, but
tryptophan.
why do you have protein, the word protein in there? Is that what it means?
Proteins do a lot of different things. Proteins are inhibitors. Proteins are carriers. Yes. What?
My question was, okay, besides proteins, you had amino acid tryptophan, why?
Because that's what it's producing.
Okay.
Its job is TRP, stands for tryptophan. Its job is to make tryptophan. When it makes tryptophan too much amount, it will then cause it to be inactivated, kind of like a feedback loop. Negative feedback.
This is so long.
The tryptophan, can we just say tryptophan binds with lactose? Is that the suppressed? No.
There's no lactose involved.
Oh, there's not?
No.
Nope.